For this topic, we will be continuing with our discussion of the digestive system. So, last topic, we discussed about the alimentary canal part of the digestive system. So that's part one. So this alimentary canal is like the main bulk of the digestive system, and it is what we think of when we say digestive system, right? Because this alimentary canal makes up the tubular structure, which extends from the mouth up to the rectum, right? And then this alimentary canal is also the main part which takes care of the ingestion of the food, mechanical breakdown, chemical breakdown, as well as the absorption of the nutrients, right? And now we will move on with the glands. So for the glands of the digestive system, we have our salivary glands, which basically help in the lubrication of the ingested food as well as plays a role in the initial breakdown of food. And then, of course, we have our liver, which is the biggest digestive gland, and then the gallbladder, which stores and concentrates the bile that is being produced by the liver. And finally, we have our pancreas, which is responsible for the production of digestive enzymes, which are secreted out in the duodenum or in the small intestines, which again helps in the digestion of food. Anyway, all of these glands are basically specialized epithelial tissue, right? If you will remember with Beta 105, um, the glands are actually specialized type of epithelial tissue. Then, your glands also contain connective tissue. This connective tissue, as with all the other organs, constitute the covering or the capsule of our glands, as well as they also form the connective tissue septa, which subdivides these glands into lobes as well as lobules. Right? Actually, um, some of the glands will also contain layers of muscle tissue, but not as extensive as and as thick as those that were found in the muscularis layer of our intestines and stomach, okay, or our tubular GI organs, okay. And some cells would also a little bit ah, I mean, it's also possible to see peripheral nervous tissue, nerve trunk fibers embedded in the connective tissue trabecula of these glands. Okay, so if we talked about the alimentary canal last time and it was tubular in basic structure, of course, for our glands, it is now they are more parenchymatous in nature. Not really more, but parenchymatous in nature, wherein they are basically grossly solid. And again, you would have your connective tissue capsule and then the subdivisions. So the capsules and subdivisions, which are made up of connective tissue, is collectively termed as your stroma. No? They do not have any of the secretory functions. They are just there to support and serve as framework of the secretory units. Okay? Okay, so what are the main functions of the digestive glands? First, it's for the lubrication of the ingesta, for its easy travel from the mouth area into the esophagus and then into the stomach. Okay? So for this, we have your salivary glands. Okay? So for most common domestic animals, we would have the major and minor salivary glands. And then this is how they look like grossly. And you can see that grossly, the um, lobes and lobules are already easily seen. Okay? So next is digestive glands would of course release enzymes. No? So the main enzyme releasing organ or gland would be our pancreas. So this is the pancreas. And again, it is still lobulated. So later when we see the histological features, we can see these lobules histologically as well. And then for the control system or control center of the digestive system, which basically functions for the metabolism of the nutrients absorbed through the intestines, as well as the storage of these nutrients and the detoxification of whatever pathogenic materials that was absorbed into the blood system, as well as the production of bile. We have the liver, right? So of course that's the liver. So it's so big because it does a lot of things, and that's why the liver is a very vital visceral organ. And it is one of the main organ that is usually affected when there is something wrong in the body because it's doing a lot of things for the body. Okay. Next, for the bile storage as well as the concentration, actually it's already here. We have our um, gallbladder, Well, aside from the storage and concentration of the bile, which is actually being produced by the liver, the the gallbladder can also produce a little bit of mucus secretion, which it also incorporates into the bile. Okay. Okay, so how do these digestive glands develop? Well, basically, actually, all of the digestive glands are just outpocketings of the gut. Okay, so for the salivary glands, they are just like outpocketings or specialized outgrowths of the foregut. And then for the midgut area, then that's where you would have the stomach. And then actually, the liver and the pancreas are just specialized outgrowths of that area as well. And what's interesting here is that you can see that it is the hepatic cords or the rows of hepatocytes that are actually developing first. Okay, and then here you can see that the pancreas actually develops from two buds, the dorsal pancreatic bud and the ventral pancreatic bud, and that these buds just eventually combine in the middle as what we see in the adult version of it. Okay. So what are the digestive glands? Again, we have our salivary glands, which have two major types, which is your major and minor salivary glands. Major just actually means the bigger salivary glands, and minor just actually means the smaller salivary glands, grossly. Okay. For major, we have your parotid, mandibular, and sublingual. And for minor, you have your labial, lingual, buccal, and then all of the salivary glands just basically mainly produce enzymes, such as your amylase and lipase. Okay? So for the digestion of your starch as well as your fat or lipids. Okay? For the pancreas, basically the pancreas has both endocrine and exocrine functions. And these endocrine and exocrine functions can be seen histologically. Specific parts would um, function for the endocrine functionality of your pancreas, and certain parts, the one that we will be mainly discussing and plays a bigger role in the digestive system would be its exocrine part. Okay, so here the pancreas is basically tubuloacinar in shape, and it produces the enzymes produced by the pancreas are basically pro enzymes in that they are not yet active when produced and still needs enterokinesis or the molecules which will activate these proenzymes by the time that they go into the intestine. Okay, so this is so that these enzymes will not digest the pancreas itself. Okay, So it needs to go first into the intestines before it is activated. Okay? An example of this is trypsinogen, which is activated into trypsin when by enterokinesis when it is already in the small intestine. For the liver, basically, the liver histologically is described or divided into lobules, one of which is histologically present. This is the classic lobule, aka the hepatic lobule. Then we have the portal lobule, somewhat imagined. Later, we'll discuss this more. And then we have the portal acynus or hepatic acynus. So these three parts or subdivisions of the liver are the main units that we have to master when we're talking about the liver histologically. And hepatic cords and hepatic sinuses would constitute these lobules. Then we have finally our gallbladder, which has the basic layers of which has a very similar layers with that of our alimentary tract or tubular part of the GIT. So you have the tunica mucosa nearest the lumen, the propria submucosa muscularis, as well as serosa or adventitia. Okay? So as a review, let's review the different types of secretory cells. So here we can see what type of secretory cell? This is actually our serous secretory cells. Serous secretory cells actually produce very thin and watery secretions. Okay? While here we have our mucous secretory cells, which produce thick and gooey or mucous secretions. Okay? So serous and mucous. So for serous cells, you would have a smaller lumen. So these are the individual serous cells, and together it will be your serous secretory unit or acynus. If there are multiple acynus or secretory units, it's called acyni. Okay? So these are your secretory units and the individual cells. So if you have round cells and more darkly staining cytoplasm, then you would have those are your serous secretory units. If you have a supposedly bigger lumen and flattened nuclei, which are seen in the periphery of the secretory units or in the basal part of each individual secretory cell, then that would be your mucous secretory units. So it makes sense because the nuclei need to be flattened in order to give space for the thicker secretory product. Okay, so they are histologically mucous secretory units are lighter and more transparent in color. Okay, and of course, we can have a combination of this serous and mucous in two ways. In one gland, your purely serous units and purely mucous units can be found in a single lobule, or in one unit, you can have a combination of serous cells as well as mucous cells. So usually, if you have a combination, the serous come in a crescent moon-shaped structure, which is termed as serous demi-lune. Lune coming from the term luna or moon because it looks like the crescent-shaped moon, no? But for me, I feel like if I would name this, I would call this Cirrus croissant because it looks like croissants to me, or Cirrus banana. Anyway, it's the same thing, no? These are basically Cirrus units, Cirrus cells combined with their mucous units. And apparently, these Cirrus demolins are actually artifacts, no? Artifacts of the um, paraffin process. Okay, if you are using the paraffin process because you would need to use alcohol chemicals which would dissolve some of the cellular components, and it would look like that the serous units have shrink into this demilume. And then scientists have found that if you would use the frozen style of sectioning, wherein you will not have to use any alcohols, these serous demilunes are absent histologically. Okay? So again, these two are the manifestations of the mixing of your serous and mucous units in your glands. Okay? What else? Aside from these, myoepithelial cells are other components that are usually seen in your glands. No, these myoepithelial cells, coming from its name, are like specialized epithelial cells which are able to contract like your muscles or your myocells or myocytes. Okay, so they are your flattened cells found at the periphery, usually of either your ducts or your actual secretory units. Okay. Usually, they are better visualized using immunohistochemistry, such as this one. So, the brown cells are your myoepithelial cells. Okay? And again, as a review, your glands can be classified structure-wise based on their ducts as well as secretory units. For the ducts, your glands can be either simple or compound. Simple if it has only one duct, and compound if the duct is branching. And then for the secretory units, if it's unbranched or branched. So this would be your unbranched secretory units, and this would be your branched secretory units. So usually, if your duct system is compound, your secretory unit is almost always branched. Okay? And then for the shape of the end pieces or the secretory units, you would have your tubular shape and your acinar or alveolar shaped secretory units. Some reference materials would actually interchange acinar with alveolar, while some would differentiate between the two. Okay? In my case, it doesn't really matter as long as they can differentiate acinar and alveolar from the tubular. Okay, but anyway, for the glands of the digestive system, it's almost always the acinar or alveolar secretory unit. Okay, that anyway, the difference between the two is that alveolar end pieces look like the alveoli of your lungs. That's why alveolar end pieces usually would have bigger lumens as compared with your acinar end pieces. Okay. So for the salivary glands, again, we said earlier that there are major and minor salivary glands. And these are the major salivary glands. And these are the minor salivary glands. And they can be further characterized by their type of secretion. Okay? As well as by their shape. And then by the method of secretion. So, for the secretion, the type of secretion, these are your serous, mucus, or mixed. So, depending on the species, this salivary glands would have different types of secretions. But the key point here is, I think, for the major salivary glands, the parotid gland is almost always serous, except for carnivores, wherein they may be slightly mixed with a little bit of mucous units. Okay? While for the mandibular, it depends on the species. Okay? But the key point here is that the animal is always right. Okay, I always emphasize this. The animal is always right. If if you see a section, for example, in rodents, but you see that it is mucus in nature, then so be it. Because the sections that are taken from the animal is what is true. Diba? Again, these references are not infallible. Okay? For the shape, all salivary glands are compound, tubulo-alveolar in shape. Okay? And for the method of secretion, all salivary glands secrete their secretions or saliva through the merocrine type of secretion. Again, there is no apical blebs nor whole cells being excreted as with your sebaceous glands. For the apical blebs, that would be seen in your sweat gland or in your mammary gland. But for the salivary glands, it's just the merocrine method. Okay? Okay, so for the basic structure of our salivary glands, we would have our secretory units, and then we would have our intercalated ducts, so smaller in lumen. And then finally, we would have your striated duct, and then your excretory duct. Bigger ducts, which would normally have striated or multiple layers of either columnar or cuboidal cells. And again, here, this would be the myoepithelial cells that we showed earlier. Okay? So in the striated duct, it is striated in nature because the cellular components of striated duct has striations or infoldings in the basal part of the cells, no? And then in every striated portion in the basal part, you would have your mitochondria, which basically gives it a darker color histologically. So here we have a section of our salivary gland, and you can see here that it is um, lobated and lobulated, okay, by connective tissue septa. And then you would have your ducts. So if the ducts are in between lobule, then it is interlobular septa. But if the duct is within the lobule, then it would be your intralobular septa. Okay? So here you can see your secretory units or acinus or acini, if there's a lot of them. And then here you will see your intercalated ducts, which have a simple cuboidal shape lining epithelium. So here, and then you would have your striated ducts, which are relatively bigger and have taller cells, so they are more columnar in nature, simple columnar. And here you can see the striations that we have talked about earlier. Okay? Here is another photo of the difference between your striated duct and the smaller intercalated ducts. So cuboidal to columnar. Okay? And again, we have here, it seems like a purely serous salivary gland, which gives us a clue that this may be taken from a parotid gland. Okay? Here, again, we have striated duct side by side with your intercalated ducts. And here, we have a portion of our interlobular ducts. So this area, and you can see here that you have a bilayer, two layers of cells. So it's already stratified. And bigger ducts would have a columnar, stratified columnar epithelium, while smaller ducts would have a bicuboidal type of epithelium. Okay, so it really differs. Usually, it is the intralobular ducts which would have smaller lumens and thus would have only bicuboidal. But if it is the interlobular, which are bigger, it's bicolumnar in nature. Okay? And you would know that these are the intralobular or interlobular instead of your striated or intercalated because they are surrounded or they are embedded in connective tissue septa, such as this one, and not directly surrounded by your acinus or secretory units. Okay? All right, so here let's see some salivary glands, and you can see that with glands that are mostly mucus in nature, it cannot still be purely mucus because here there are still darkly staining areas or acinus or secretory units which are serous in nature. So if you consider the whole gland, it is still mixed in nature. Okay? While here we have a what looks like a purely serous gland in general because of the dark staining, and we don't see much um, lighter staining structures. Okay. Here we see a mixed gland again because this area contains all of the mucous secretory units, while this area contains the serous secretory units. Again, here, what we already discussed this earlier. We have our serous units, darkly staining, round nuclei, mucous secretory units, lightly staining, flattened, peripherally located nuclei. Okay? Again, this is so because mucus is thicker, it needs bigger lumen and more space for the storage within the cells. Okay? Next, let's go to the pancreas. So again, the pancreas this time, I don't know why, but most references would say that the uh, salivary glands are tubuloalveolar, while the pancreas is tubuloacinar. But histologically, later if we look at it, it actually looks the same. For me, it can be interchanged. Okay, again, pancreas, especially the exocrine part, is the one that plays a bigger role in the digestive system, okay? Because it produces proenzymes. So first, let's talk about the exocrine part of it. So for the exocrine part, we would have our pancreatic secretory unit made up of the pancreatic acinar cells as well as your centroacinar cells, which actually are just a continuation of the ducts, okay? So as with your salivary glands, you also have your intercalated and intralobular ducts. Okay? What else? In each of these pancreatic acinar cells, we would see the zymogen granules that are located in the apical portion of the cells. Okay. Here is a, this time, here is a another schematic diagram. So you can see the pancreatic acinus exocrine function, and then here you will see a group of endocrine cells. Okay, that's why the pancreas has exocrine and endocrine functions. Right. So as a review, exocrine glands have ducts. Its secretions pass through ducts in order to go to the area where the secretions are needed. While in endocrine glands, they don't have ducts and their secretions are mostly hormones which are expressed directly via diffusion into the bloodstream. Okay? Anyway, here you can see that the pancreas kind of looks like the salivary gland as well, but there are major differences. Anyway, we can see here that the um, pancreatic secretory glands look like they are serous in nature, right? Which makes sense because pancreatic juices are not thick, they are watery. Okay? So here you can see the lobations as well, the pancreatic acini and the ducts, and then the islet of Langerhans. A closer look into the pancreatic acini, you would have your acinar cells and then your centroacinar cells. Okay, your centroacinar cells actually are the ones producing the bicarbonates, which a little bit neutralizes the high pH that is being produced by the high pH of the of the pancreatic juices being produced by the acinar cells. Okay. So here, again, we have our acinar cells and then the centroacinar cells. And then here, in this photo, we can see that the zymogen granules or the little packages of pancreatic juices are still seen in the apical portion of your acinar cells. Okay? For the endocrine, we have our pancreatic islet, and the pancreatic islet is actually composed of several types of cells which are basically indistinguishable from one another under H&E, but um, the most famous are your glucagon producing cells, which are your alpha cells, and insulin producing cells, which are your beta cells. But if we have the time and the resources, we can always use IHC. So here you can see you would have your beta cells and alpha cells. So again, this is immunohistochemistry.